So thank you so much for the invitation to speak. I hope everybody can hear me. Um, can you hear me?
Yes,
Okay,
we
excellent,
can
great.
hear you great.
So thank you for that really kind introduction and also for the opportunity to speak at this great symposium. Um, I'm going to talk today about some of our recent work in understanding gene regulation in health and disease using single cell genomics and really building improved maps of human tissues across a diversity of conditions and data that could be used for improving machine learning models, AI models, and so forth, as well as some of the insights that we've gained into the biology of specific diseases. So in particular, our group is very interested in what drives gene regulation, and this is largely through what are called cis-regulatory elements. These are short sequences that are located throughout the human genome and are responsible for regulating the activity of genes and dictating the identity and function of human cell types. And so every cell type will have a different combination of cis-regulatory elements that it uses to regulate gene activity. There's a variety of sequence-based assays that we use to annotate cis-regulatory elements, attack-seq, chip-seq techniques that most of you have probably heard of, if not very familiar with. In particular, the technique that we use oftentimes is attack-seq. And what attack-seq does is exploits a phenomenon of gene regulation in that sequences are typically wrapped around nucleosomes and inactive. And in order for the sequences to be used by regulatory proteins to drive gene expression, nucleosomes have to be displaced or removed. And that exposes the underlying sequence, allowing regulatory proteins to bind to drive gene expression. And this is called open or accessible chromatin. And attack-seq exploits this phenomenon basically by integrating sequencing primers into these regions of open chromatin. And you can sequence the results of these assays, and that allows you to map the locations of these elements, for example, promoters or distal elements such as enhancers. And so, of course, we're using single cell assays to annotate cis-regulatory elements in human tissues. And given that human tissues are heterogeneous, this allows you to understand the gene regulatory programs and regulatory elements in individual cell types. And I'm going to skip over the details of how we go from tissue to clustering and annotated cell types, given that I presume the audience is fairly familiar with that. What I will touch on a little bit is the types of assays that we use to map gene regulatory logic. So as I mentioned, single cell versions of attack-seq, as well as increasingly paired attack-seq and RNA-seq generated from the same cell, and RNA-seq to measure gene expression. We're also increasingly doing newer assays, for example, an assay called paired tag that was developed in Bing Ren's lab at UCSD, where you can map the location of histone modifications in RNA-seq from the same cell, as well as Hi-C, which is a technique to map interactions between regulatory elements, particularly those that are located far away from the genes that they're regulating. And so in our work, again, this is a major focus, is mapping cis-regulatory elements and gene regulatory programs. And as one example, a previous study that we performed that was published a couple of years ago, we mapped cis-regulatory elements in the pancreas and blood. And so on the left here is showing you the clustering of all these cells that we generate in these experiments, and they're colored by the cell type annotation. From attack-seq data, we annotate cell type identity based on, since we don't have gene expression from these assays, we annotate it based on the activity of the gene promoters that are known to mark certain cell types, or in some cases throughout the gene body of these genes. And in doing that, that allows us to, in this case, identify pancreatic and blood cell types. We could then take the profiles from each cell type and then define cis-regulatory elements within the cell type. And in this study, we identified about 450,000 elements across all cell types. And I've just highlighted here a couple examples, some of which are very specific to cell types. So CFTR is a marker of pancreatic ductal cells, and there are cis-regulatory elements, a bit hard to see on this image, that are very specific to ductal cells relative to other cell types. More broadly, we took these 450,000 elements and asked which are very specific to a cell type relative to others. And about 5% of these elements were highly specific to a cell type. And if you take those elements and ask what types of genes they're near, they tend to be located near genes that are involved in highly specialized cell type functions, arguing that these cell type specific cis-regulatory elements are those that are dictating the highly specialized functions of each cell type. In another example, we've looked more broadly across many different human tissues. And so this was a study that was done in collaboration with Bing Ren at UCSD, where they looked at 45 adult and fetal tissues and performed a TAC-seq and all these tissues and identified over 200 different cell types, adult and fetal cell types, and then mapped over 2 million cis-regulatory elements across all cell types together. And again, categorized creeds based on their specificity as well as sharing in this study across similar cell types within different tissues. So one of the key ways that we like to use information from these experiments is to interface with human genetic data. And this is because variants that are associated with genetic risk of complex diseases and traits are mostly non-coding and mapped to regulatory sequences. And so we can intersect data from GWAS studies with cis-regulatory elements from cell type specific maps to understand not only the location of cis-regulatory elements within these cell types, but which of these elements is potentially playing causal roles in the development of disease. So in this example here, this element overlaps a variant that's associated with disease, and therefore the regulation of a gene through this element may be on the causal pathway to disease. And combining GWAS with these cell type specific maps can help us understand elements, genes and pathways, et cetera, that are involved in risk of disease, as well as the context. So when and where these genes are affected in response to certain stimuli, in certain developmental conditions, for example, as well as the direction of effect on disease. Does increased or decreased activity of a site and a gene lead to increased or decreased risk of disease? And we have many examples of utilizing these maps in combination with genetic data to annotate elements that might be involved in disease. Here's an example for type 1 diabetes that's upstream of this gene CFTR, which is, again, a marker of ductal cells, also the gene that's mutated in cystic fibrosis, harbors a variant that's associated with type 1 diabetes, and therefore regulation of CFTR via this element is likely playing a causal role in the development of type 1 diabetes. We've, again, done this more broadly across many different tissues and cell types where you can perform analyses to identify first, broadly, which cell types harbor risk of certain traits and diseases. And this allows you to annotate traits that match up with certain cell types to understand the cell types involved in these traits and diseases. So here are some examples of that, as well as, again, annotate specific loci for these diseases in terms of the cell types they're affecting, as well as the regulatory programs that these variants are impacting to potentially cause disease. So based on this introduction, and one limitation, I would say, of many of our previous studies, several which I just described, are that we've generated what I would call largely reference maps from fairly modest sample sizes. And so the question that we're interested in is how can we build on these studies to gain deeper insight into the role of human cell types and physiology and disease, moving beyond just reference maps of each cell type. And so to do this, we've been performing expanded single cell profiling of donors from both healthy and diseased states. And that allows us to ask a variety of questions, such as what's the extent of normal variability in the regulatory programs of human cell types? How do these programs change during disease? When combined with genetic data, what changes during disease progression or physiology are causally involved in disease? Increasingly generating data from not just TAC-seq and RNA-seq, but multiple layers of the epigenome and gene regulation. And so how does layering multiple levels of data inform these insights? And then finally, this gives us a greater diversity of cells, cell states, phenotypes from donors, and just greater variability that is useful for informing the development of models, just as David talked about previously, using machine learning and AI to make predictions from these data. And so in our studies, I'm not going to talk about all these today, but I just highlight that we are performing expanded studies of a variety of human tissues in both non-disease and disease states. Pancreas, liver, heart, lung, and brain, just to name several of the ongoing studies that we have. And then I also wanted to note that these studies are done with a variety of collaborators. Our closest collaborators are Bing Ren, who's my upstairs neighbor, as well as Alan, who runs the Center for Epigenomics at UCSD, where a lot of these data are generated, and we work on these studies together. And then we have a variety of other collaborators for each of these different projects. One thing I wanted to note about these studies that we've been doing to expand the number of donors that we're able to profile in single cell studies is that we've been increasingly using study designs that allow us to generate data from larger numbers of samples while keeping the cost as low as possible. And the way that we do these studies increasingly is to, instead of running individual assays per sample, we pool tissue from multiple donors, and then we extract nuclei from the pool, and then we run assays serially on this pooled donors. We also genotype the donors before they're pooled, so that when we recover cells from the pooled experiments, we can match up the identity of the cell to the donor it came from, based on matching the genotype of the cell to the genotypes of the donor. And so that gives us donor-specific profiles starting from sample pools. And some of the advantages of doing this are that it's cost-effective per sample. It allows us to calibrate cell numbers per donor. It also has a lot of advantages in terms of doublet removal, because most doublets are mixtures of multiple individuals. And it also gives you a more uniform background that is easier to account for when you're analyzing, say, differences in control and disease, as opposed to doing individual assays per donor. Okay, so I'll talk mostly today about our work in the pancreas, which was our primary interest in our lab, specifically is pancreas and its role in diabetes. And I'll also talk a bit about another study we are doing in the liver. In the pancreas, there are three things I want to touch on in terms of how this expanded profiling has informed our understanding of the pancreas and how it's regulated and its role in disease. One is by looking at variability in regulation across non-diabetic individuals, so in physiology. Two is changes that are occurring in type 2 diabetes progression, and three, changes that occur during progression to type 1 diabetes. So for the first study here, so this is a paper that recently posted on bioRxiv that's in review, where we profiled a single-cell multiome, so again, paired ATAC-seq and RNA-seq of individuals who were non-diabetic, so didn't have disease, and defined gene regulatory programs in each of the cell types in the pancreas. And so one thing to note here is that these are data from pancreatic islets, which are endocrine cells that secrete hormones such as insulin that are isolated from the pancreas. So the majority of cells in these data are endocrine cells, alpha and beta cells, although there's a variety of other cell types in the pancreas as well. And so we define the identity of these cell types based on the expression of genes, and then we can also confirm that these same clusters have accessibility from ATAC-seq at the promoters of these same genes. And again, we take these profiles from each cell type, and then we define cis regulatory elements within each cell type. And in this study, there were about 300,000 total cis regulatory elements that we identified across all cell types. So one of the questions that we're interested in is just what's the extent of variability in the regulation of these cell types in the non-disease state. And so these are showing some insights into how much variability exists in these measurements across donors. So on the left here is the abundance of different cell types. So there is quite a bit of variability in the abundance of different cell types. Some of that is technical due to how the cells are isolated, but a lot of that is also likely, some is also sampling. variability, but likely some of this is also biological in terms of differences in cell proportions in individuals, and actually can be quite extreme donor to donor. On the right, if we look at the variability on the top here, the measurement of gene expression, and the bottom measurements of accessible chromatin at specified genes. For gene expression, in many of these genes, there's almost an order of magnitude difference in expression of these genes across individuals, again, all in non-disease states. We see less variability in the ATAC-seq, but still there is some degree of variability. And so both in terms of expression and chromatin profiles, we see wide variability donor to donor in these regulatory programs. And so what we can do is use this variability, we can leverage it to understand associations with different phenotypes as well as genetic variation genotypes that we're collecting on each of these donors. And so the way that this works is that we take our single cell map, and then we have the regulatory programs of each individual cell type, and then we can ask whether certain phenotypes are associated with regulatory programs in each cell types, as well as the genotypes of genetic variants across the genome. And so I'll start first by describing briefly the results for phenotypic associations. And so what we found, which is quite interesting, is that, first of all, there was quite extensive associations with different phenotypes that we had available for these donors, again, in non-disease state. But what was particularly interesting is that within a cell type, there were highly distinct responses of the same cell type to different phenotypes. So, for example, in beta cells, so the beta cells are the cells that secrete insulin, and they're the cells that are most important in diabetes. There was almost no overlap in the pathways in this case that were associated with each of the phenotypes that we tested here: age, sex, BMI, and this is glycated hemoglobin levels, HbA1c. So there's quite diverse responses of cell types to different phenotypes, even in the non-disease state. And I've highlighted here a couple pathways that were associated with variability in age, in this example, in different cell types. And in beta cells, we saw a significant upregulation of pathways related to hypoxia and cell death, as well as downregulation of pathways that relate to unfolded protein response as a mechanism that the cell uses to deal with stress. And so downregulation of that process is maybe also contributing to dysfunction of beta cells. Even though these aren't disease states, age and BMI and these other factors are risk factors for the development, particularly for type 2 diabetes. And so understanding normal variability in these programs across these phenotypes can give us some insight, not only into just how cells can vary across these different dimensions, but also how this may contribute to disease in older individuals or heavier individuals. We also looked at epigenomic drivers of these cell types, so same phenotypes, but now looking at associations with sequence motifs that are enriched in the accessible chromatin profiles of each cell type. And what was noteworthy here is that a lot of the factors that we find associated with these phenotypes are external responsive factors. So they're responding to external signals. And in the case of age, there's a set of factors that we identified that are associated with age in beta cells that respond to circadian rhythms, respond to other external signals, or nuclear hormone receptors, which are responsive to hormones. And that suggests that the changes within beta cells in age, and BMI is similar, may be due to external drivers of these changes, as well as likely some intrinsic changes that are occurring in the cells as well. Okay, so we also, so that's phenotypic association. So we also mapped genotypic associations for these same cell types. And here, and these are called QTLs, quantitative trait loci. So we identified about 8,000 QTLs across all the cell types. Many of these are shared across cell types, so the 20% are either cell type specific or lineage specific, and these QTLs disrupt different transcription factors that themselves have either cell type or lineage specific activity. And on the right here is one example of a cell type specific QTL. So you can see the genotypes of this variant here, the activity of this cis regulatory element in beta cells was dependent on the genotypes of this variant, whereas in these other cell types, the activity doesn't change as a function of genotype, right? So the effects of this variant were highly specific to beta cells relative to other cell types. And one of the ways that we use this information for QTLs is to ask for variants that affect gene regulation in these cell types, do these same variants affect risk of developing diabetes and or related traits? And so here's one example of a locus where variants affect the activity of a site in beta cells that's predicted to regulate this gene DYRK3. The same variants are associated with type 1 diabetes. The protective alleles for type 1 diabetes increase the activity of the site. And so that suggests that decreased activity increases type 1 diabetes risk. And that may give us clues as to a mechanism how these variants operate, and that this element and gene may be involved in risk of type 1 diabetes in beta cells. So that's changes in physiology. So we find extensive changes in the non-disease state, although some of these then do tell you something about how physiology may contribute to diabetes. But as I mentioned before, we're also looking at changes that are occurring in people who are actually progressing to disease. And so very briefly, I'll touch on this study, which was published in Nature Genetics last year, where we looked at progression to type 2 diabetes. So individuals could be normal glucose tolerant or have type 2 diabetes. And then there's a state that's known as pre-diabetes, where you have impaired glucose tolerance, where you're not diabetic, but you have reduced beta cell function. And it's oftentimes a precursor to developing type 2 diabetes. And so for this study, we profiled the pancreas using a TAC-seq and RNA-seq of non-diabetic, type 2 diabetic, as well as pre-diabetic individuals. And so here are the results of clustering these data. This is the TAC-seq profile. So we find same alpha, beta, other endocrine cell types, as well as other cell types in the pancreas, and can then ask questions about changes that are occurring in type 2 diabetes. And one way that we use these data, which I thought was relevant to the symposium, is that so we were interested in the question of whether these pre-diabetic cells had intermediate genomic phenotypes compared to non-diabetic and type 2 diabetic donors. And to ask this question, what we did is to train a classifier to basically allocate the cells to either non-diabetic pre-type 2 diabetic or type 2 diabetic states, and then ask whether the cells mapped in different classes, especially for pre-T2D, or whether the cells mapped in only the non-diabetic and type 2 diabetic state. And this would support models in the former, where if they were mapping to a third state, that these cells were intermediate, distinct enough from non-diabetic and T2D donors. But what we found is actually that instead of having intermediate phenotypes, the pre-type 2 diabetic cells either map to non-diabetic or type 2 diabetic states, but just in different proportions. So this argues that there's intermediate proportions in pre-disease, not intermediate genomic profiles, whereby an increasing proportion of cells have diabetic-like properties. And it's the number of cells that you have in these states that may be pushing people towards the development of type 2 diabetes. And you can see this here, the changing proportions of cells that are classified as non-diabetic or diabetic, and pre-T2D is intermediate, as well as on the bottom here, where there's a significant difference in an intermediate proportion in the pre-type 2 diabetic state compared to non-diabetic and type 2 diabetic. Okay, so cognizant of time here. So, okay. So then that's type 2 diabetes. We also are very interested in type 1 diabetes and understanding changes in gene regulation and progression to type 1 diabetes. So in this study, we profiled pancreas tissue from individuals, again, who were non-disease or who had type 1 diabetes, and then also individuals who were positive for autoantibodies. So this is a biomarker of future development of type 1 diabetes. It's often seen, not in everybody, but in many individuals, that can be used to identify people who are at risk for future development of type 1 diabetes. And so we did profiling of non-disease, autoantibody positive, so like kind of, it's not pre-disease, but somewhat of a pre-disease state, as well as people who had both recent onset type 1 diabetes and long-term type 1 diabetes, and asked how gene regulation changes across this axis. And so this is the map here where we have clusters of each cell type in the pancreas from hundreds of thousands of cells. And as with the other studies, we define cis-regulatory elements in each cell type. So what we did in this study was to ask whether there were differences in the expression of genes as well as cis-regulatory elements, and then also across entire pathways in each cell type in people who are either type 1 diabetic or autoantibody positive compared to non-disease individuals. And what this is showing here is the number of pathways that he had significant up or down regulation in each of these comparisons. And just to highlight one example of this, so in beta cells, in individuals who recently developed type 1 diabetes, we see upregulation of pathways related to interferon signaling. So the cells are being destroyed by the immune system, and therefore, not surprisingly, we see responses to immune signals in beta cells, as well as presentation of antigens and other immune-related processes as well. But we were quite interested in not just looking at individuals who had type 1 diabetes, but what was going on in the preceding states and people who are autoantibody positive to gain insight into what changes might be occurring before disease. And just at a high level, I'll say that we find some similarities in the preceding stages. So the pathways I just mentioned, cytokine signaling, interferon signaling, antigen presentation, are all upregulated even in the early stages of disease. But there are other pathways that are either not changed in autoantibody positive donors compared to T1D, as well as pathways that are only altered in autoantibody positive donors compared to T1D, suggesting that some of the changes that are occurring are intermediate compared to future development of disease, as well as processes that are completely distinct that are only altered in these pre-disease states that you don't see necessarily changes in disease. And this highlights that, first of all, it's valuable to study donors in pre-disease states because you gain different insights into people who had disease, where you may be observing a lot of consequences of disease, and also gives us some insight into processes that may be, driving progression of an individual to disease. That's gene expression. We also see similar changes in the epigenome. So certain transcription factors have intermediate activity in the pre-disease state, whereas others have changes only in the autoantibody state compared to type 1 diabetes at the motif level, and then also at individual cis-regulatory elements, many of which are predicted to regulate genes that have changes in expression. And these elements in particular are quite interesting to us because they may be targets for epigenome editing to potentially protect cells from destruction in type 1 diabetes to, in these cases, dampen or silence the activity of these sites to reduce the expression of these genes to mirror what the activity of these sites is like in the non-disease state. Lastly, for this part, so these are changes that we're observing in progression to disease, but we don't necessarily know which of these changes is causally involved in disease. And so again, this is where we use human genetic data to try to distinguish causal versus consequential processes. And in this case, we took the pathways that were changing in progression of disease and asked whether cis-regulatory elements regulating these pathways preferentially harbored genetic risk of, in this case, type 1 diabetes. And so here we find across all these pathways that are changing in disease, a subset of these pathways are harboring disease risk variants preferentially, whereas all these other processes aren't. So I always like to highlight this one example of apoptosis. So beta cell death is often used as, it's the endpoint of disease in type 1 diabetes. And it's often used in genetic studies to, as like a readout to understand genetic variant function. But apoptosis, it doesn't actually appear to harbor much risk of type 1 diabetes, and therefore beta cell death is more likely to be consequential of disease as opposed to driving disease risk. And some of these processes here I've highlighted on the top are likely more involved in disease causing events. Okay, I think I'll skip over this part in the interest of time. Yeah, I just quickly, we're very interested in following up on these studies and mostly profiling larger numbers of donors and then subsetting them and increasingly smaller bins. And in this study here, we're looking at non-diabetic donors preferentially, and then asking what changes are occurring in the very, very early stages of disease, and using genetic risk to separate high versus low risk individuals, which has revealed some processes that are altered only in high risk individuals versus low risk individuals, and may give us insights into why some people go on to develop diabetes and why some don't. Okay, but the last part of the talk, I'm going to touch on studies in a different tissue, which is the liver. Similar concept in that we're looking at changes that are occurring in liver cell types in non-disease and disease individual. But in this case, we're looking at what's called metabolic dysfunction associated steatotic liver disease. That's a mouthful, also known as fatty liver disease, which is an extremely common disease. So it's estimated to affect, which is really an unbelievable number, about 30% of all adults in the entire world. And there's different stages of MASLD. Some are not particularly severe, so steatosis or MASLD, whereas MASH is a more severe form of the disease. And the changes that are occurring in progression to advanced disease are really not well understood, nor are the causal pathways that well understood. And there's a real unmet need for therapies to be able to treat MASLD, which, as you can see, affects a substantial number of people in the world. So that's what we're aiming to get at in this study, which is single cell profiling of the liver in non-disease and MAFLD. And so here we have about 90 donors across non-disease and different levels of severity of having MAFLD, where we do single cell multiome assays, as well as paired tag, histone, and gene expression, and droplet Hi-C, which are 3D interactions, as well as genotyping on all 90 donors. So these are the results of the single cell map. And this is just multiome data, so over 400,000 cells. And we identify the major liver cell types, annotated again based on gene expression, and we identified substantially more cis-regulatory elements in the liver than have been reported in previous studies. We also identified quite extensive heterogeneity if you subcluster these cell types. Has been reported in many contexts previously. So in hepatocytes, there's a lot of zonation of hepatocytes based on where they're located in the liver, and they have distinct markers, as well as particular subsets of disease-associated hepatocytes, such as senescent cells and stress-associated cells, and we can identify all of these populations. We also map the paired tag data as well to the same level of resolution, which gives us, in this case, K27 acetyl is an active mark. K27Me3 should be there, is a repressive mark, sorry. And that allows us to annotate the activity of these elements that we identified from the multiome map. Are they activating elements, or are they repressive elements? And we have the same resolution of cell types from these data to be able to make those annotations. So we look at changes in abundance of cell types on the left, as well as cell subtypes on the right. And just to say that there's extensive associations and changes across progression to disease, including cell types that increase in abundance, as well as cell types and states that have decreased abundance. We also find quite dramatic changes in gene regulation in terms of expression and chromatin in MASH, which is the more advanced form of the disease. By comparison, very few individual changes in MASLD, which is the steatosis, the very early stage of disease. So it's a more mild form of the disease, and we don't find many changes. And if we ask the question if there's a correlation in the effects of changes in genes in the less severe and the more severe form of the disease, for many cell types, we find strong correlation suggesting that the less severe form of the disease just has an intermediate phenotype. But in hepatocytes, there's almost no correlation in the changes in MASH and the changes in MASL, which is quite interesting. And if we look at pathway enrichments of the association results, we find that very different pathways that are enriched in the less severe form of the disease versus the severe form of the disease, suggesting that, again, it's not an intermediate in terms of progression from less severe to more severe disease. It's actually distinct, pretty substantially so, in terms of its gene regulatory programs, both in terms of pathways as well as sequence motifs quite dramatically different in the less severe, more severe forms of the disease. Okay, let's see where I am on time. Okay, so we then take the cis-regulatory elements and then ask whether they're enriched for genetic association for mast cell, which gives us insight into the cell types that are driving disease. So hepatocytes show strong enrichment, as well as the cell type cholangiocytes, which are biliary cells that also show strong enrichment. And if we subset the sites within these cell types based on those that have up or down regulated activity, in progression of disease, hepatocytes are enriched sites that go down in disease, whereas the other cell types are enriched for risk in the sites that go up in progression of disease. So this allows us to filter down to subsets of sites that may be driving disease risk, not only the cell types themselves involved in disease, but the particular changes in these cells that are disease relevant. And so finally, for this section, since we have genotype data for all these donors and all these modalities, we can map genetic effects on each of these pieces of information, expression, chromatin, and then histone modifications. And we have done this, and this is the number of QTLs that we find for each of these modalities, largely in the more common cell types like hepatocytes, but we find QTLs in other cell types as well. And as I described for diabetes, we can use this information to map up to risk variants associated with liver disease and annotate genes that are involved in liver disease risk. And so in this case, this locus is associated with liver disease, the expression of this gene EFHD1, as well as accessible chromatin and histone modifications, all in hepatocytes, all in the same direction. So this suggests that by layering QTLs with disease risk signal, we can gain insight into not only mechanisms of disease, but potential novel targets for treating fatty liver disease. Okay, so I'm going to skip that part. So I'll summarize the talk. So I think the overall summary is that expanded profiling of gene regulation and specific cell types in human tissues in both health and disease states gives us greater insight than just looking at reference maps of small numbers of samples. So there's extensive variability in associations and regulation, and even in non-disease and physiology. Pre-disease states have distinct profiles compared to disease. Sometimes it's intermediate, but more often these are actually completely distinct processes than we see in disease states, and highlights pathways involved in progression to disease that may be therapeutically actionable. Finally, genetic risk of disease, largely mediated through gene regulation. We can identify cell types involved in disease, as well as pathways, genes, and so forth that are on the causal path to disease. And I'll just end by saying that given the theme of the meeting, that really this diversity of data from individual cells, from different tissues and different donors and disease states, and oftentimes rich metadata that we have for these donors, overall will be really informative for the types of models that people are building using machine learning and AI to make predictions and understand these diseases. So thank you very much. Again, I'm thrilled to have been invited to speak, and really it's a great symposium, and I'm very happy to answer any questions. So thank you.